Hello everyone, this is Lucky Limbu from 9xwood and today we are going to discuss about C3 cycle, or you can call it Kelvin cycle. So if you are having problem regarding C3 cycle, particularly to memorize its compounds and the bulky reactions, obviously this video is for you. So before I start, you should have a basic knowledge about photosynthesis, about the light phase reactions. If you remember the light phase reactions, there are the products ATP and NADPS2. So which simply means adenosine triphosphate and nicotinamide adenine dinucleotide phosphate hydrogens. So these are the products of the light phase reactions, and the C3 cycle, which is a dark phase reactions, these products are going to be used. So to simplify the process, there are only three steps you have to remember. So they are, first one is the carbogylation, second one is your phosphorylation, third one is your reduction. If you just can remember just these elements, half of your cycle is completed. So let's take to these carboxylations. Carboxylations, by the words, means that additions of the carbon dioxide, and phosphorylations means additions of an inorganic phosphate. By the name, phosphorylation means addition of the inorganic phosphate. So whenever you are doing phosphorylations, this simply means that you are converting adenosine triphosphate to adenosine diphosphate. So if you see here, this triphosphate is being converted into diphosphate, which simply means that one inorganic phosphate is being released. So this one inorganic phosphate will attach to the substrate you are using and it will be phosphorylated. Next one is the reduction. In the reduction, this simply means that addition of the hydrogen. So reduction means addition of hydrogen, and when you are reducing anything, this simply means that NADPS2 is being converted into NADP. So this releases hydrogen, and this is going to be reducing agent. So these three are the basic elements, and if you can remember these three elements, half of a cycle is completed. Now this first stable compound 3-phosphoglyceric acid will undergo phosphorylations. As I have already told you, phosphorylations is nothing but the conversion of ATP into ADP. And this will release one inorganic phosphate, because triphosphate is being converted into diphosphate, and this inorganic phosphate will be attached to this substrate. And if you see here, these two molecules are being released. So we need 2 molecules of ATP and 2 molecules of ADP, and this will results in, since already one phosphate group is here, adding another phosphate group will result into biphosphate. So the product will be 1,3-diphosphoglyceric acid, and this is 3 carbon compound, and this is also 3 carbon compound. So the next process is reduction. Reduction simply means addition of the NADP so as to release NaS2 as NADP-S2 into NADP. So this will release one hydrogen, and this hydrogen acts as a reducing agent, right? So as it is also being released in the 2 molecules form, so we need 2 molecules of NADP-S2 and 2 molecules of NADP. So it will reduce this compound, and as acid is being reduced, we will get aldehyde, which is phosphoglycid aldehyde, and it is also 3 carbon compound, so we write here 3, or you can remember like 3-pagal. Since the compound 3-PGAL, 3-phosphoglycid aldehyde, is a member of a carbonyl compound, we have 2 members of the carbonyl compound, they are aldehyde and the ketone. So it will isomerize into ketone, and if you remember here, 2 molecules of PGA undergo phosphorylations to give diphosphoglyceric acid, which is also 2 molecules, and it is being reduced, it will also be 2 molecules. So 1 molecule of the 3-phosphoglycid aldehyde isomerize, we write this symbol for the isomerizations, to give dihydroxyacetone phosphate. This is aldehyde, this is ketone, and they are isomer to each other, and 1 molecule is being isomerized to here, and 1 molecule is being left here. but the fact is that these reactions are happening in the stroma of the chloroplast within a fraction of seconds. So it's quite inconvenient to say that one molecule, two molecule, but for simplicity we can use the term. So 1 molecules of the phosphoric acid aldehyde isomerized to give ketone, right? And now this 1 molecule left and combined with this acetone to give you fructose 1,6 diphosphate. So it is an 6 carbon compound. Now this formed compound fructose 1,6 diphosphate is very near to the glucose. So if you remember, the whole process of Kelvin cycle was to generate glucose, and fructose is also a 6 carbon compound, so does glucose is. So now we have to just do few steps to release glucose. So here it is diphosphate, but we don't need a diphosphate, we need a phosphate. So to remove phosphate, we use phosphatase enzyme. If you remember, for adding phosphate we use ATP. For removing phosphate, now you can remember phosphatase. So it will yield us fructose 6 phosphate. Now this fructose 6 phosphate will be converted into glucose, which is the main need of the Kelvin cycle. So up to here you see, first is the carboxylations, adding of the carbon dioxide, phosphoric acid, first spiral compound, it undergo phosphorylations and to give diphosphoric acid, undergo reductions to give phosphoric acid aldehyde, isomerase to give acetone, ketone, and now they combine to give fructose 1,6 diphosphate, which one gives glucose. So up to here, from this conversion of phosphoric acid aldehyde to glucose, what we are basically doing is reduction. So we call this process as reduction process, this whole process as reduction process, or you can also call it glycostatic reversal, because the reactions is reversed, the same reaction is reversed in the glycolysis of the respiration. And since this is a cycle, Kelvin cycle, half of the portion is completed up to here. To complete the cycle, fructose and phosphoric acid aldehyde will combine to give you erythose, which is a 4 carbon compound, and it will give you, after several reactions through many enzymes, it will give you pseudoheptulase, which is a 7 carbon compound, which in reactions will give you ribulose monophosphate. And to regenerate RUBP we need biphosphate. So to convert monophosphate into biphosphate, we just need to add a 1 phosphate group, which can be achieved by simply adding a ATP. It will be converted into the ADP, releasing a phosphate group, and this phosphate group will combine to this monophosphate, and thus RUBP can be regenerated. So carboxylation, reduction and regeneration are the basically 3 elements of the Kelvin cycle, and it is also releasing us glucose. At the same time, it is also regenerating RUBP. But if you see here, the process up to from the conversion of fructose 6 phosphate and the phosphoric acid aldehyde to regenerate RUBP seems very simple, but it is not the fact. In fact, it's a multi-step, and it is quite hard to learn all this, but I will make sure that it will be as easy as I have read it. So let's go to this regeneration process. For the regenerations of RUBP, which stands for ribulose biphosphate, and to complete the cycle, you just have to remember 2 things. Whenever you see 3-PGAL, this means whenever you see 3 phosphoric acid aldehyde in the reaction, just write transketolase. Whenever you see 3 phosphoric acid aldehyde in the reaction, write trans ketolase enzyme over the reactions. It will be easy for you. And whenever you see dihydroxyacetone phosphate, whenever there is a reaction between any compounds and the dihydroxyacetone phosphate, just write aldolase. Just write aldolase over here. Or you can also remember like this, as this dihydroxyacetone phosphate is a ketone, and ketone gives aldol condensations, if you remember chemistry. This is the two things you have to remember, that phosphoric acid aldehyde use enzyme transketolase, for dihydroxyacetone phosphate use enzyme aldolase. So let's begin the cycle to regenerate RBP. The first process is the reaction between fructose 6 phosphate with phosphoglyceraldehyde in the presence of enzyme transketolase. So it will give you erythrose 4 phosphate along with xylulose 5 phosphate. So if you see here, this xylulose is a 5 carbon compound, and this ribulose biphosphate is also a 5 carbon compound. So it can easily isomerize to give RBP, and the regeneration process can be completed. But we cannot ignore the fact that erythrose 4 phosphate, a 4 carbon compound, is also being produced. So we have to also convert this product into 5 carbon compound. Then only the regenerations will be effective, right? So the next reaction is between erythrose 4 phosphate with dihydroxyacetone phosphate. So what is the enzyme going to be used? That is aldolase, because dihydroxyacetone phosphate is used, aldolase. So it will give you seroheptulase 1,7 diphosphate. So if you see here, this is a 7 carbon compound. You can also remember it as hepto means 7. So it is a diphosphate, but we don't need diphosphate, we just need a phosphate molecule. So it undergoes a process to remove this phosphate. So to remove this phosphate, an enzyme phosphatase act over it, phosphatase. So it will convert it into seroheptulase 7 phosphate. Now this seroheptulase 7 phosphate again combines with this phosphoric acid aldehyde in the third reaction, and this seroheptulase 7 phosphate react with your phosphoric acid aldehyde in the presence of the enzyme transketolase. So it will give you seroheptulase 7 phosphate and phosphoric acid aldehyde in the presence of the enzyme transketolase will give you xylulose 5 phosphate plus ribose 5 phosphate, right? So both of which are 5 carbon compound, and now the process of isomerism become easy. And in the fourth step, this xylulose 5 phosphate now isomerize. For isomerize we use this symbol to give ribose 5 phosphate, and this is ribose 5 phosphate, and we need to regenerate ribulose 5 phosphate. So we are very near to the conversion. So now this ribose 5 phosphate will again isomerize to give ribulose 5 phosphate, and we need ribulose biphosphate. So we need to add a phosphate group. How can we add the phosphate group? By phosphorylations. So the final and last step is ribulose 5-phosphate converting into ribulose biphosphate by phosphorylation. So in this final step of phosphorylation, ATP is converted into ADP. Thus it releases a inorganic phosphate group, and this inorganic phosphate will be combined to here. So it will give us ribulose 1,5-diphosphate, which is RUBP. In this way, RUBP is regenerated, and it will now again start another Kelvin cycle. So in this way, one Kelvin cycle is completed. First proceeding with the carbonylation, and next is the reduction, or you can call it glycosidic reversal, and next is the regeneration, the RBP. So if you see here, in one Kelvin cycle, just one carbon dioxide is fixed. So it is used in the process of making glucose, so which is a 6 carbon compound. Glucose being 6 carbon compound, and at a time just one carbon compound is fixed. So we need 6 cycles for the formation of one glucose. So if you observe this, that in one cycle 2 ATP and here 1 ATP is used. So total 3 ATP. So we can write here in one cycle 3 ATP, and how many NADPS2? NADPS2, 2 NADPS2 is used. So for one cycle 2 NADP is required to produce one glucose. So for the formations of the one glucose, that means for 6 cycle we need 6 into 3, 18 ATP molecules, and 6 into 2, 12 NADPS2, and this is what the Kelvin has found. And Kelvin and his co-workers have also calculated that for one cycle it requires just 15 seconds, and for 6 cycle it would be 90 seconds. So this was for the Kelvin cycle, and I hope you enjoyed the video.